Hi everyone. Right, this is very weird for me. So here's where we left off. We'd been through, I think we had been through all of the limiting factors for photosynthesis. We did talk about light. Now I think that we got to the point where I was talking about enzymes and the fact that this is an enzyme controlled reaction. And so with that in mind, just trying to see if I can see my cursor. Yep. Everything that affects enzyme activity will affect how well photosynthesis is working. So temperature, the same applies. It will increase, the rate of photosynthesis will increase as temperature increases until we get to the optimal temperature. And then after that point, we start to see denaturation of the enzymes that are involved in photosynthesis, and it will eventually cease working. Can you think of the name of an enzyme that was involved in photosynthesis? I'll give you a sec. So hopefully you might have come up with this one right here, ATP synthase, an enzyme involved in photosynthesis. And then, of course, any of the inputs will be a limiting factor. So if there's not sufficient carbon dioxide, then photosynthesis won't be able to take place. If there's not sufficient water, which is an input of which stage of photosynthesis? If you said light dependent, then you are correct. All right, so we can move on then with the last part of chapter 5, looking at cellular respiration now. There's a lot of similarities between photosynthesis and cellular respiration, so I'll keep referring back to the bits of photosynthesis that we can draw a comparison to. But just starting with your key knowledge, so the purpose of cellular respiration, why does the body or a plant undergo cellular respiration? What's the goal? What's it achieving? The location, so much like photosynthesis, we want the location. We want to know all about that mitochondria, the parts of it, and where each of the stages of cellular respiration is happening. So yep, the mitochondria is the site of cellular respiration. The structure and the evidence of their bacterial origins. So remember that endosymbiotic theory that applied to chloroplasts, the same story for mitochondria. And the main inputs and outputs of each of the stages of cellular respiration. All right, we need to touch on anaerobic respiration first. So anaerobic respiration is distinct from cellular respiration, although they do share one stage in common. So anaerobic respiration is what will happen in the absence of sufficient oxygen. There's 2 stages to anaerobic respiration. Glycolysis is our first stage. In glycolysis, which I'll go through in a little bit more detail in a sec, in glycolysis we're effectively splitting a glucose molecule, and that will obtain a net amount of 2 ATP, because it's a catabolic reaction. If we're breaking the bonds that are holding that glucose molecule together, in breaking those bonds you're going to release that energy. So that energy is then used to rebuild that cycling coenzyme, ATP, which we would have started with ADP. With the addition of an extra phosphate, then we end up with our ATP. So glycolysis is giving us 2 ATP. It's then a bit of a choose your own adventure. So if there is insufficient insufficient oxygen, then we will follow the anaerobic respiration pathway and fermentation will take place. The things we need to know about fermentation mainly is what the outcome is. So in animals, if fermentation takes place, that's when we get lactic acid produced. So if you're taking PE, then you'll know all about this buildup of lactic acid and how that can affect your muscle function. If we're dealing with a plant, yeast, or bacteria, then the product is ethanol, an alcohol, which you can smell. The point of fermentation, so fermentation itself doesn't yield any extra ATP than what we obtained in glycolysis. The point of fermentation is to free up that cycling coenzyme that we see used here, NAD+. A distinction that VCAR draws usually is that fermentation, when they say the word fermentation, they're actually referring to anaerobic respiration. So if you're asked how much ATP is produced in fermentation, the answer is 2. Even though fermentation itself doesn't produce any further ATP, it includes the process of glycolysis. So we have 2 ATP being produced in fermentation. Right. And so the diagram, which I'm covering here, aren't I? Let me move me out of the way. The diagram that you can see to the side here shows us the process, and this is from your textbook, but it shows us how we're starting with that carbohydrate, that monosaccharide glucose, and in splitting that molecule, that energy is harnessed to create 2 ATP. Without oxygen, this pathway here, we're going to follow fermentation, which gives us our products at the bottom. You don't need to memorize these biochemical changes. You don't need to memorize these structures, but it's just handy to see it, I think, represented in a diagram. If after glycolysis has taken place, we have sufficient oxygen, then the body will follow the cellular respiration pathway. Okay, getting to know our organelle that we're dealing with here. So the mitochondria, which that's the one that we spoke about in class before we wrapped up, saying that when you did your project back in year 8 on the cell and you made your little jelly beans, the mitochondria, and you would have had to make a label that went along with it, and it would have said something like, it is the powerhouse of the cell. So that's the last time I ever want you to say that the mitochondria is the powerhouse of the cell, because we have to put some better bio terms to it. So instead of saying the mitochondria is the powerhouse of the cell, we can refer to it as being the site of cellular respiration. You can elaborate further and explain that cellular respiration is the act of creating ATP, harnessing the energy from a glucose molecule to reload and make usable ATP. So this is the mitochondria, the organelle that we're dealing with, and you've got to be able to recognize it in a few different representations. So on this slide, you can see those different representations for you. So if it's in a diagram, you should be able to clearly see the double membrane structure. We've got the outer membrane, and that folded inner membrane kind of looks like a deflated balloon inside another balloon. If you're looking at a real life image, an electron micrograph, then it has a bit more of I guess, a rigid structure. And you can see how it can be represented in a few different shapes, but still looking for those folded structures inside the football or jelly bean shape organelle. So I've pointed out the inner and outer membrane. The folds that I referred to just now of the inner membrane, they have a special name. They are called cristae. The reason or the importance of having cristae as part of the mitochondria structure is that it increases surface area. I might come back to that point when we get up to a few slides' time, because that increased surface area, one of our processes for cellular respiration is taking place on this inner membrane. So the more surface area we can have of that membrane, the more enzymes that can be available for that process to take place. So we'll come back to the importance of the cristae later. And then just mentioning again that the mitochondria have their own DNA. So mitochondrial DNA, it's a circular piece of DNA, which again is testament to its origins. So much like bacteria have that circular double-stranded piece of DNA, mitochondria have their own as well, further evidence that they were their own organism before they were engulfed by the developing eukaryotic cell. And just one more label that you'll want to pop onto your summary sheet. So I can just remind you of, I can work out how to get out of the slideshow. Yep. Remind you that you've got this cellular respiration summary sheet, like we had for photosynthesis. You've got your mitochondria there. So you'll want to be labeling your outer membrane, your inner membrane. You can label these as cristae. And then my favorite part of the mitochondria, we enter the matrix. So the middle of the mitochondria there you can label as the matrix. And just to remind yourself where we are positioned, we're in a cell, so the outer environment is going to be the correct cytoplasm. Okay, before we go to the next slide, you can hit pause for a second, and what I want you to do is try to recall the overall chemical reaction for yourself for cellular respiration. It's something that you have learned in the past, no doubt. Let's hit pause and see how close you can get to the actual overall reaction. How well do you know it already? Go. Okay, so hopefully you had a go for yourself, and let's see how close you were. Ta-da! All right, so like I explained with photosynthesis, cellular respiration isn't just one reaction that's taking place. It is the summary of, in this case, 3 different reactions that are happening. Actually, a lot more than that. 3 different stages that have so many different reactions happening as part of each stage. So it's a summary reaction of everything that's going on in this whole process. But that is the accepted overall equation that you just need to commit to memory. You can see, and we already talked about in class, that it is sort of the backwards of photosynthesis. Instead of our glucose being an output, it's now an input. Likewise with oxygen, and the reverse is true for our water, where we had more going into photosynthesis. We've now got it only as an output for cellular respiration, and carbon dioxide being an output as well. We've now got this addition of our cycling coenzyme ATP. And we talked already about how the number has changed in recent times, and even when you were learning it in your junior years, you might have recorded it as 36 to 38 ATP, whereas it now is more accepted to write 30 to 32. It is the general feeling that VCAR won't discriminate on the amount of ATP that you write. So if we can just we'll pick the number and stick to writing 30 to 32 ATP. But if you find yourself in that panic moment forgetting exactly how much ATP that was, it won't be something that will lose you marks. And we've got our 3 stages that we're going to run through. So glycolysis is occurring in 3 stages. Cellular respiration is occurring in 3 stages. Glycolysis, which we already spoke about, the Krebs cycle, and the electron transport chain. And just summarizing there how much ATP is produced in each stage. So pretty much glycolysis in the Krebs cycle is preparing our molecule. We're getting a little bit of ATP from that process. but it's preparing our molecule for that final stage where all of that ATP is going to be generated. Because the ATP is the purpose of cellular respiration. Being a key knowledge dot point, I guess we should talk about that one for a minute. When we were running through photosynthesis, I brought up the idea that that between the two stages of photosynthesis, we have a lot of ATP being produced in the light-dependent phase, and then that ATP is used up to create this glucose molecule. So what's the importance of that glucose molecule? For a plant, it is transportable. That glucose molecule can now be transported down to a root cell, which needs ATP. It needs energy for growth and function. So here's where we're going to use that glucose molecule, take all of the energy that's stored in the bonds that hold it together, and build the energy into the bonds of our ATP so it can be readily used. We're getting these ready to use little packets of energy for the cell, whatever cell we're talking about. All right, you've got here a link to an interactive, internet-based interactive, which works to take you through the different stages of cellular respiration. I'm just going to hit pause for a second, make sure the link is working and show you it quickly. All right, there we go. So if you click that link, it will take you to this interactive, and it's separated out into 4 videos. The big picture talks about glycolysis cellular respiration as an overall reaction. Glycolysis, little video will take you through. So I don't know if you guys could hear that playing, but if you follow the link yourself, you'll be able to work through each of the interactives and hopefully get an understanding of how cellular respiration is taking place in its 3 stages. I think do that first before you keep playing this video, and it might make a bit more sense. All right, so here's where you want to play along with your summary page. So starting with glycolysis, let's try to show on your summary page where it's taking place first of all. So if we have a look on this diagram, glycolysis, you want to pick somewhere sort of outside the cell. Might work from left to right, so I guess I'd put it up here somewhere. And just showing that breakdown of the glucose molecule and the idea that we have 2 options. So an arrow to show the direction of the pathway if we have insufficient oxygen, being the anaerobic respiration pathway and some detail of fermentation. And then if we do have, if there's enough oxygen present, and you just label it with O2 present, then your arrow is going to shoot across here and head into the matrix. But just talking about glycolysis for a sec, let's have a look back at the slide. And you want to get the information that you need to fill into this stage here. So you should have your overall reaction written up here. Number 1 is going to be glycolysis, and the inputs and outputs will take you through now. All right, so we're in the cytoplasm. Input is glucose. Outputs would be 2 ATP. The interactive might have you confused because it does talk about the 4 ATP being produced. Don't forget that as the input for this phase, we had 2 ATP being used. So you can just leave it as a net production of 2 ATP. We end up with the glucose molecule that's now been split in half. Each one is referred to a pyruvate molecule, as a pyruvate molecule. So we have 2 pyruvate and 2 NADH. And it sounds familiar from photosynthesis. We've talked about all three of our cycling coenzymes now. So ATP, where we're starting with ADP, and if we load on that extra phosphate, we get ATP. NADPH was talked about in photosynthesis. So NADP is the unloaded form, NADPH is the loaded form. I like to remember that it is P for photosynthesis. So that might work for you. And now we've got our third cycling coenzyme, NADH, so no P. And this one is strictly used in cellular respiration. So the unloaded form of NADH is NAD+, and when it's loaded, it's NADH. So an output of glycolysis is NADH, two of them. And there'll be more to come as we head towards the electron transport chain. Because we've got loaded cycling coenzymes as outputs, you can include their unloaded forms as inputs. So to your input list, you can add ADP and NAD+, being the unloaded forms. Okay, once we've got our 2-pyruvate molecules, we've had a couple of bonds already broken in the glucose molecule, and that energy has been used to reload and store energy in ATP already. I might have to tend to something in a second, hang on. Okay, the cat's rubbing the screen. You want to do some photosynthesis? You want to say hi? Meow. He's hungry. I'll hit pause. I'll be back. All the children are in bed now. So we have our pyruvate molecules now with all that energy still stored in the remaining bonds. Those 2-pyruvate, now if we've got enough oxygen, will migrate into the matrix of the mitochondria. All right, now if you've done the interactive, your mind's probably racing with all these things that were happening as part of what we call the Krebs cycle. The good news is that you can leave it as a summary of inputs and outputs, so what went into it and what came out of it. The rest of it, you just need to understand, is a series of chemical reactions that take place, lots of bonds being created and broken down, and eventually what we get at the end is a whole lot of NADH. There's this other one that we call FADH. It is because NADH is the only one that is mentioned in the key knowledge in the study design. We only really ever talk about NADH, but FADH serves much the same purpose, so it's worth mentioning. ATP, and that's where we get our carbon dioxide being produced as well. So inputs for the Krebs cycle we want to list as 2-pyruvate. Once those pyruvate come in and start the Krebs cycle, the action of the Krebs cycle is just to make sure that the energy that's contained in those remaining bonds is used as efficiently as possible to build more ATP and NADH. The carbon dioxide that gets produced, where is that going to end up? Where's it going to go? It is an overall output of cellular respiration. So when you're labeling it on your little summary sheet, it's destined to leave the mitochondria, leave the cell. If it's happening in your body, where does the carbon dioxide end up going? Just take a deep breath in and then let it out, and there goes all your carbon dioxide. So it's going to leave the cell, enter the bloodstream, be picked up by your red blood cells, and you're going to eventually get rid of it through your respiratory system. All right. So doing a quick check, we've so far of our ATP collection, we've achieved 2 in glycolysis and 2 in the Krebs cycle. We've talked about our overall output of carbon dioxide, and we've collected a whole lot of these things called NADH along the way. All right. And here is where the action happens. So last stage of cellular respiration, called the ETC, the electron transport chain. All right, this is what I was referring to earlier as the stage that's taking place on the cristae of the mitochondria. So this explains why it is important to have all that increased surface area, because the more inner membrane we have, the more of these transmembrane proteins and enzymes we can have, and the more electron transport chain activity can be taking place. All right, so here's how it works. And hopefully you've done the interactive, because it's quite useful for understanding this stage in particular. So we have our NADH, and I'll only talk about NADH, but FADH does the same thing. The NADH molecules arrive at the electron transport chain. We have these things called electron pumps. They are transmembrane enzymes that are doing a job, and our NADH is going to drop off 2 things. So they're referred to as electron carriers. They are delivering their electrons to these electron pumps. The energy from those electrons is used to take this hydrogen that they were also carrying and pump it across into that intermembrane space. So in between the inner and outer membranes. If we have a look at your summary sheet, I think a good place to pop your Krebs cycle diagram is probably here, where there's a little bit more space. And then choosing a place like, say, over here, where you can put down some detail of the electron transport chain. You want to have your 3 embedded electron pumps, and then that last enzyme that I'll talk about in a minute can go here. All right, and so we're going to show that NADH has lost its hydrogen, and that hydrogen's being pumped across into the intermembrane space, so that we end up with a really high concentration of hydrogen sitting in here. Okay, so there's all our hydrogen sitting in the intermembrane space. It's called an electron transport chain because there's multiple electron pumps that work one after another.
I found this on the web.
Siri's just trying to help. And the electrons move from the first pump to the next one, pump more hydrogen through, and then to the last pump, pumping more hydrogen through, and then they'll sit there. And I'll come back to them in a minute, because they're just going to sit there and wait for the last piece of the puzzle to happen. All right, so our high concentration of hydrogen ions, and then this embedded enzyme here, which hopefully this scenario is looking a little bit familiar, because it should look like something that we've already gone through. It should look very much like, have a guess? It should look like the light dependent phase of photosynthesis. So where we had the water splitting to form that big concentration of hydrogen ions inside the thylakoid. Now we've got this high concentration here of hydrogen ions in the intermembrane space, and they're going to want to move from the high concentration to the low concentration. So we're going to have this flow of hydrogen ions going through this enzyme, and we're already familiar with this enzyme because it is, again, ATP synthase. And the job of ATP synthase? Building ATP, which is the end game now for cellular respiration. So our hydrogen ions will move back through ATP synthase, And while they do that, we're going to have our ADP coming in, much like we had for photosynthesis. ADP coming in, you can draw that little curvy arrow to show ADP on this side, and then coming out the other side we have ATP. How much ATP? For the overall reaction, specifically for the electron transport chain, we should have about 28 to 30 ATP being produced. Okay. So we talked about our inputs, NADH, FADH, which again, it's not in the key knowledge, only NADH is really mentioned, and the fact that we're producing this ATP at the end. What we haven't mentioned are our two other big players. So oxygen being an overall input for cellular respiration, we haven't addressed that one yet, and our water molecules that are coming out as products. So how do these two fit into the puzzle? This very last little action that happens here. So the electrons that have moved along the pump, they will sit tight on that last electron pump until they are collected by an oxygen molecule. So when you're asked, or if you ever asked in an exam question, say, what is the role of oxygen in cellular respiration? The standard line is, it is the final acceptor of electrons in the electron transport chain. So oxygen collects its electrons because that would make it fairly negative. It will also collect some hydrogen ions. And what do you get when you mix hydrogen and oxygen? There's our H2O molecules being formed. So we have oxygen coming in, which will leave as water molecules. And that, my loves, is cellular respiration. So just having a quick look back at your summary sheet. Hopefully now you've got a nice summary of cellular respiration on this document with a neat list of inputs and outputs, which when it boils down to your knowledge, this is what you need to know. These are the things that you need to be able to commit to memory. Being able to see how they fit into the whole process and where things are happening is, my husband just walked past, so this is a bit awkward. Seeing how they all fit together in the mitochondria and how they each play a part, that will help you to have an overall understanding of the process and be able to recall the little bits and pieces of information, like what is the importance of oxygen, what is the importance of the cristae in the mitochondria. I hope I didn't waffle on too much, and I hope this was helpful. If you have any questions, email me, and I'm looking forward to see your Chapter 5 summaries. Chapter 5 summaries probably should just be made up of perhaps a 1-page of summary notes and your summary sheet. So if you can attach your photosynthesis and your cellular respiration, these summary sheets, that is pretty much what your Chapter 5 summary should consist of. See you soon.